Alright guys, welcome to Module 7, Parasitology, Virology, and Mycology. Module 7 is going to focus on mycology. So again, here are some objectives for you guys to go ahead and go through and be able to answer. Most of the questions for the final and for this week's quiz are going to be found in this lecture specifically. Most of the answers are going to be found on the slide, and you'll get some of it from my actual lecture. Alright, so one thing I wanted to note about this lecture is I went through and put most of the important information right on the slide. So a lot of this lecture is going to be kind of me reading off the slides and adding a little bit of interjection here and there. But that's because I thought it would be a lot easier for you guys to just page through the PowerPoint and look for the specific slides to get the answers. Again, I wanted to note that most of the time, in general, for a medical lab technician with mycology, you just need to be able to recognize that a fungus is present or a yeast is present, and then from there it just goes on to get treated. For the purpose of your boards, you need a little bit more information, which you'll get from this lecture, but beyond that, you just need to be able to recognize that a fungus is present. So mycology is the study of fungi. This group of organisms includes yeast, which are unicellular organisms, and molds, which are multicellular filamentous organisms. So molds frequently tend to be the source of annoyance when they contaminate laboratory media, they spoil food, or they destroy items made of wood or other fibrous materials. And then yeast has been around for centuries, and it's used for the production of food and beverages. So some of the yeast, such as Saccharomyces cerevisiae, is known as baker's yeast, and this is known to raise bread dough through fermentation of carbohydrates and production of carbon dioxide. So we have fungus and we have yeast and molds that fall into the fungus family, but not all of them are bad. So fungus are found everywhere, and they tend to thrive in moist environments. So they play an important role in returning nutrients to the soil by contributing to the decomposition of animal and vegetable matter. And then there are about 150 species that can cause human disease. In this lecture, we're going to cover a little bit of terminology, etiological or causative agents of fungal diseases, and we're going to cover mycoses, which is a fungal infection. So when we observe a fungus under a microscope, we kind of get the impression that fungi are kind of small plants because they look like really small plants or small trees. Early classification of fungi, they were thought to be part of the plant kingdom, but they're not plants because they lack chlorophyll. They're heterotrophic. They have cell walls made of chitin. So fungi are eukaryotic organisms, so they have a true nucleus bound to a nuclear membrane. They have mitochondria, endoplasmic reticulum, and ADS ribosomes. So plants are autotrophic and they contain chlorophyll, and they obtain nutrients by absorbing inorganic compounds through photosynthesis. But fungi lack chlorophyll and they're heterotrophic, so they require performed organic compounds. So fungi will secrete extracellular enzymes into the environment to break macromolecules into their subunits, and then they absorb these subunits. And then these cell walls of plants are composed of cellulose, whereas fungi cell walls are formed from chitin. Most will grow well at room air. Some of them are facultative anaerobes, so that means that they grow under anaerobic conditions. They do not require increased levels of CO2 as some bacteria do to grow, but they do prefer a moist environment. They tolerate a wide range of acidic or alkaline conditions. When they can grow in They can grow in environments with pH values from anywhere from 2 to 10, but neutral pH tends to be the best growth environment for fungi. Typically, they can grow under refrigeration from 0 to 8 degrees Celsius to temperatures as high as 45 degrees Celsius, but optimal growth temperatures for fungi are 25 to 37 degrees Celsius. Now, in the laboratory, we're going to use 37 degrees for our incubators. So, a clinical laboratory that's trying to grow up a fungi will use a 30-degree Celsius incubator for fungal cultures. So the medical importance of fungi. So some of the diseases that you guys may have heard of that are caused by fungus are vaginal yeast infections, diaper rash, athlete's foot. You could also ringworm is caused by a fungus, and then fungi don't usually infect healthy individuals. They're typically going to go after somebody who has a predisposed condition that allows them to be infected with a fungal infection. So somebody with metabolic imbalances such as diabetes, or somebody who's pregnant, somebody who has a debilitating infection such as HIV, someone with a neoplastic disease such as leukemia, lymphoma, or sarcoma, somebody who is on immunosuppression with corticosteroids or cytotoxic drugs, and then somebody who may be getting radiation therapy or administration of long-term broad-spectrum antibiotics. So these conditions make it more likely for somebody to develop an infection with fungi. A lot of the times you may read about somebody who's had an HIV infection that progressed to AIDS, and then that patient dies from something like a cryptococcus infection. So cryptococcus is one of the most prevalent fungal infections in somebody with HIV that causes death. But in a healthy individual, it may not progress that far. So when I took mycology, one of the things that was hardest for me to wrap my mind around was the structure of molds and some of the terminology. And so I'm going to spend a little bit of time going through the slide and maybe adding a little bit of interjection here. So the body or thallus is composed of a long tube-like hyphae that produce a fuzzy or fluffy colony. There are hyphae that are septated and have cross walls at regular intervals, and then they have some that are aseptate or non-septate hyphae and have no or very few cross walls. Mycelium is the mass that develops as the hyphae grow. Vegetative mycelium anchor the mold colony. Aerial mycelium supports reproductive structures and provide the texture of the colony. So a mycologist or somebody who studies fungi specifically is going to rely heavily on macroscopic and microscopic morphology of the vegetative and reproductive structures to identify a mold. Um, So molds are multicellular, filamentous form of the fungi are often described as fuzzy or fluffy. So the body of molds can be referred to as a thallus, and it's composed of a long cylindrical or tube-like structures called hyphae. So I'm just going to draw a little bit of a structure here. This would be considered like a hyphae. So the diameter of a hyphae could range anywhere from 3 to 20 microns or micrometers. So then a hyphae could have a septate, and they contain cross walls at regular intervals, which separate the sections into tubular growth. So a septate is basically just going to be a cross wall, and they're going to be at a very regular interval. So you'll have to excuse my drawing here, but this would be uh, a hyphae that has septate. So this is a septate, this is a septate, and then this whole structure is a hyphae. And you'll get to see that on some of the slides as we move on and progress. So there's multiple uh, types of hyphae. So again, there's aseptate or non-septate and then septated hyphae. Both types of hyphae elongate growth from the tip of the atypical zone. Mycelium is the term applied to the mass that develops as the hyphae grow. branch and intertwine. So the whole structure itself is going to be referred to as a mycelium. Hyphae penetrate the surface of a growth medium in order to absorb nutrients of the vegetative mycelium. Um, so some of the reproductive structures you might see typically is just going to be on, you're going to have a hyphae here, and then it's just going to have like a little projection on the ends, and that's going to be the reproductive structure. And then some molds are referred to as damacious molds, and they have dark pigmented hyphae due to the presence of melanin-like pigment. So these hyphae are going to be a little bit darker, and when they're darker, they're considered damacious. Again, that's here. Highland molds are non-pigmented. They begin as white, and they reproduce color as the reproductive structures mature. Claf Other fungal elements include rhizoids or stolons, and these are found in like zygomyces or Rhizopus species. So they're kind of like root-like structures, and we'll cover that as we get into further into the lectures. So in the last few slides, we talked about the structures of molds, but now we're going to talk about the structures of yeast. So yeast are a form of fungus. They're unicellular in nature. So as they grow on a solid media growth, they form a discrete, round, smooth, moist, or mucoid colony similar to that of bacteria. These colonies are composed of spherical to ellipsoid cells that range in diameter from 3 to 15 microns. And yeast are actually able to produce asexually by budding or fission. So most of you guys may have seen budding yeast on a urine slide, or you've heard about it already in any of the our classes, and that's actually that yeast is reproducing and forming another cell. So during the budding process, a swelling appears along the cell wall of a mother cell as a result of a weakening of the small portion of the cell wall. The nucleus of the mother cell divides by mitosis, and as a result, the cell now contains two nuclei. As the swelling continues to enlarge cytoplasm, and one of the nuclei follows into the daughter cell, and eventually a septum forms between the mother and the parent cell, or the parent cell and the daughter cell. Eventually, separation of the two cells occurs, leaving a budding scar that is clearly visible on an electron micrograph, and the cell production is a budding uh, process called blastocondium or bud. On some occasions, two cells elongate but remain connected, continuing to divide until a chain of blastoconda uh, is produced, forming a filamentous structure referred to as a pseudohyphae, and this can be seen in Candida tropicalis species. Pseudohyphae differ from true hyphae as a constriction is apparent in the common septum between the parent or mother cell and the daughter cell. So basically, a blastoconda is going to look like a hyphae, uh, but there's a big difference because it's not one actual fungi, it's going to be multiple that are just connected together. Some yeast cells can also produce a filament, which is a growth or a growth at the tip. And there's going to be no constriction at the origin from the parent cell, so there's not going to be any separation from the parent cell and the daughter cell. And this is known as a germ tube, and it could be useful in identification of Candida species. So most of you guys have heard of yeast infections, and most of them are going to be coming from the Candida species. Germ tubes eventually become true hyphae as septate develop along the length of the filament. Identification of yeast species is usually accomplished through a combination of biochemical testing and an observation of a microscopic morphology to determine the fungal elements. Some fungal species are able to grow in two different forms, exhibiting both yeast and filamentous mold phases. They're known as dimorphic fungi. So when we think about fungi, there's going to be some fungi that only exist in the hyphae form or the mycelium, and then there's going to be fungi who only exist in the budding form or single cell form, but then there are also some species that are going to be dimorphic and they have both the yeast and the mold phases. We could use brain heart infusion auger with the sheep blood and incubate it at 35 to 37 degrees Celsius to grow some yeast forms. Yep. Asexual reproduction. Sorry, guys, Amber stopped by, so I had to pause. But asexual reproduction in yeast cells results in the formation of blastocandida, and in the filamentous molds result in the formation of some type of condidum or candida. Candida can survive harsh environmental conditions such as dry atmospheres and produce as a result of mitosis. They're known as asexual spores, and they usually produce on the tips of specialized aerial hyphae called condidophores. And you would see this with Aspergillus species. Basically, what it is you see the hyphae and it branches out into like a flower projection at the end. With a lot of the candida resting on the top. So it essentially just looks like a flower. And then the Aspergillus species is where it's represented. So you would just see like little circular projections all throughout here. I'm trying to draw it, but it's not working. Um, so this condium or spore form at the top of bottle-shaped lides, and they sit on a large end of the stalk like condidophore. Septate hyphae, so those are the ones that have like the septates in them, uh, enlarge and separate to become arthrocandida, and then round chlamydocandida is the large resting spore that can form at the end of hyphae on the terminal end or with the hyphae in the intercalary. So sexual reproduction, I think, hold on, I got kind of lost here. So in zygomyces, asexual spores are formed within sac-like structure, and these sac-like structures are called sporangium. So specialized aerial hyphae called sporangiophores bear the sporangium containing sporangiospores, and they may be branched or unbranched. And what it is a round, sterile supporting structure called columella is formed at the tip of the sporangiophore and extends into the sporangium and serves to separate sporulating or non-sporulating areas. So it's sexual production. So we just covered asexual reproduction. Now we're going to cover sexual reproduction. So sexual reproduction involves the union of two haploid, also known as N, nuclei, each possessing a single set of chromosomes to form a zygote, having a diploid, which is 2N, nucleus with two sets of matched chromosomes. So meiosis or reproductive division then follows, producing four haploid nuclei. So to avoid confusion, it should be noted that the sexual form is called a telomorph and the asexual form is called an anamorph. Also important to note that sexual reproduction has not been demonstrated in all fungal species. Those species that do not undergo sexual reproduction, as well as those species for which the sexual stage has not been demonstrated, are classified as fungi imperfecta. Most of the human pathogens belong to this group. Demonstration of the sexual stage allows other species to be placed into the classes Ascomyces, Blastidomyces, and Zygomyces, based on their morphological characteristics of their sexual stages. We rarely see the sexual stage in a clinical laboratory because the two compatible isolates are required and identifications usually involve only one mating type. There are some clinically important species that are sexually self-fertile, and no discussion of sexual reproduction would be complete without the description of the structures involved. Um, let's go into the next slide. So the Ascomyces include fungi whose sexual spores are encased in sac-like structures called an ascus. These spores are called ascospores, and they can be seen with the Saccharomyces species. The Blastodomyces include fungi whose sexual spores and blastospores are borne on minute stalk-like structures called stigmata. Mushrooms are known for the presence of this structure. Blastodomyces create blastospores. Oh, okay, sorry, I already covered that. Zygomyces form thick-walled zygosporum containing a single zygospore, and that's how they reproduce. Sexual reproduction is not too important, but it's also important enough for us to go through it because it helps us kind of further classify these uh, fungi so that we could identify them. This is just a quick note here, safety measures in handling fungus cultures. We're going to want to use a biological safety cabinet, and what this is going to do is it's going to prevent us from being exposed to spores. So we're going to jump right into the saprophytic fungi. Fungi can abandon their saprophytic existence to cause opportunistic infections with those who have HIV, leukemia, lymphoma, cancer, a transplanted organ, diabetes, and pregnancy. The most common species are Aspergillus and Candida. And what they do is they invade weakened host defenses, grow at body temperature, and they exist at the low partial pressure of oxygen that occurs in host tissues. So, um, like fuzz that appears on food that's stored under refrigerated conditions after long-term storage, they're more or less considered saprophytic fungi. The fungus that you see growing on bread also would be considered saprophytic. Excuse me. Opportunistic infections caused by saprophytes, so aspergillosis. It's caused by Aspergillus fundamentis, and it's the most common species that causes aspergillosis infections. So how we would diagnose these, and most commonly what we're going to do in the lab is we're going to do a potassium hydroxide wet prep or direct examination. And what we're going to do here is we're going to use potassium hydroxide, which is a cellular clearing agent, and then a gram stain. And and what we're going to look for is the presence of fungal elements and that could be associated with aspergilli. Most fungal elements will be seen as gram-positive, um, whereas some mucus and host tissues in that preparation is going to appear pink. Um, Again, a lot of the times we're not going to do the fungus cultures, but it's important to note what these colonies it might look like. So, with Aspergillus species, the colony is going to be rapid growing, so anywhere from 2 to 6 days. The colonies are going to appear yellow to green, to dark green, green or blue, or have a black surface. They're going to appear velvety or have a cottony surface. And at the end of this lecture, I have pictures that are going to represent this. You can kind of see what these colonies are going to look like on a growth. And then the reverse of the colony is often white or lightly colored. With Aspergillus, we're going to see a hyaline uh, with septated hyphae. It's the condylophore terminates in the swollen vesicle, Phyllidates cover the entire vesicle of varying degrees on it, and then chains around smooth of rough candida. So, what for us, most likely what we're going to want to look for is the presence of the septated hyphae. So here's a colony of Aspergillus fung, fumigatus, and it's a dark green with a velvety-like appearance. And then here's Aspergillus galaticus. We see the oval vesicle on the top of the stalk-like condylophore with its bottle-shaped phyllodates. The numerous round condida resting on the top of the phyllodates, and they're kind of slightly rough. So this would be considered a Aspergillus species, and then this would be the reproductive spores at the end. So fungi in the class Zygomycetes have also been implicated as etiological agents of human disease. They include the genre Rhizopus, Mucor, Rhizomucor, and Absidia. The term zygomycosis includes clinical condition known as mucormycosis, and predisposing factors for infection with these are somebody who is under immunosuppression, somebody who may have lymphoma, leukemia, malnourishment, metabolic disorders, and trauma. Mucormycosis refers to a disease caused by Rhizopus or mucor and rarely Absidia species. The normal habitat for these fungal species in soil includes soil or decaying matter. In a human host, they are capable of causing several disease states, including renal, cerebral, pulmonary, gastrointestinal, subcutaneous, and disseminated disease. Most infections happen from inhalation of spores, and the consequences, nasal sinuses and lungs, are the most common sites of initial infection. So rhinocerebral mucormycosis is the clinical form most frequently encountered. We're going to see this with Rhizopus rhyzis. It's an opportunist and it's a major etiological agent. So somebody who has predisposing risk factors has enhanced susceptibility to this disease, and those factors include acute diabetes, hyperglycemia, metabolic ketoacidosis, and that happens from uncontrolled diabetes, and then leukopenia. So what happens is the infection begins in the turbinates and nasal sinuses, and then has a tendency to spread to the orbit of the eye, the palate, and eventually into the brain, where cranial nerves are destroyed. If it's left untreated, this disease can rapidly progress, may end with coma and death, occurring in less than 1 week time. This fungus exhibits preference for blood vessels, so vascular invasion and leads to thrombosis or blood clots, could lead to infarction and tissue necrosis. Early symptoms of this infection would be a headache, nasal congestion, pain, serious black or blood-tinged nasal discharge, and we could see periorbital swelling. This is a picture of a mycorrhizomycosis that is spread to the eyes. Here we're going to diagnose by a direct examination. So again, we're going to do our KOH wet prep. We're going to see aseptate hyphae. So there's going to be no septate in those hyphae or branch forms. They're broad with a right angle branching. They may appear wide like a ribbon that tends to fold onto itself, and they will break easily. Here, what we're going to want to do is we're going to grind the tissue. Grinding tissue can decrease viability of the organism, and for growing on the colony, they grow within 1 to 4 days at 30 degrees Celsius, and they produce a white colony that turns gray when it matures. Uh, Personally, I've only ever done wet preps. I've never grown a fungal culture, so I wouldn't spend too much time trying to understand how to grow these. As long as you could recognize what genus or species causes what types of disease, and then have a general understanding of the morphological characteristics under a microscope, then you should be fine. So when we look at these species under a microscope, the rhizopus are species is going to be unbranched, sporangiophore with a rhizoid at the base, and either single or in clusters. The sporangium is round and dark pigmented with lemon-shaped sporangiospores. Abscidida has branching sporangiophores arising from the stolon located between the rhizoids, not directly on them. We're going to see pear-shaped sporangium with round to oval sporangiophores. If you look in your Coleman's Colored Atlas, you may find some pictures at the back that can show you what these fungi actually look like. With the microscopic examination of the Mycology species, it's going to show branching sporangiophores, it's going to lack rhizoids, and it's going to show round sporangium with oval sporangiospores. So this is the Rhizopus species. There's a root-like rhizoids as well as aseptated hyphae are easily seen. So you see there's no septate in the hyphae here. The sporangioform at the end releases the sporangiospores. This is the area where this fungus is going to replicate. And this is the rhizis. It's a root-like thing at the end of the species. So there's many different species of fungus that have been associated with mycotic keratitis, which is a fungal invasion of the corneal epithelium. But the most common in the United States is Fusarium solani. So Fusarium species are often noted for their pink or lilac colonies. The color varies with the species, and these organisms produce hyaline septated hyphae with large curved canoe or banana-shaped multicellular microcondylata at the end, or right angle phylidates. This organism can also produce clusters or single-celled microconda or short phylidates. So here's the fusiform species. You see we have a curved macrocondylata at the end that resemble canoes. Um, you can't really see the septated hyphae in this photo, but I think if you could note the canoe-like structure and classify it with the Fusarium species, you'll be fine. So other genres that are associated with mycotic keratitis is going to be Aspergillus, Acrominium, Pseudodalsariboides, Candida albicans, Dermaceous fungi such as Bipolaris, Curvulvera, and Cladiosporum species. Predisposing factors is accidental ocular trauma, surgical ocular trauma, ocular tropical corticosteroids, or antibiotics. It's most often seen with farmers, fruit pickers, and gardeners. This is the bipolar species that causes this disease. The macrocondylata are large with thick-walled and multicellular. They create slight bends where they attach. So you see these really slight bends where they're attaching there. This is a Curvulvera species with multicellular, thick wall. Note the center is larger than the others. You could see the septates here in the large center. A lot of times with your boards, you're gonna get a clinical disease and you're gonna get a picture and they're gonna ask you what fungi is causing this. I had a lot of mycology on my boards, which I'm still surprised that I passed them because I've had so much. So there's going to be some initial discomfort with a foreign object on the surface of the eye, which is followed by pain that intensifies over the next several days. Redness of the eye is readily apparent, and examination by the clinician will reveal the corneal ulcer surrounded by a white, gray, or yellowish corneal infiltrate. Also, you could also see satellite lesions with hypopyon, which is a collection of pus at the anterior chamber of the eye. Without treatment, loss of the eye is inevitable. This is known as mycotic keratitis, the disease we're talking about. The next disease we're going to talk about is mycotic otitis. So saprophytic fungi may invade the outer ear, leading to another opportunistic infection known as mycotic otitis. This is going to be caused by Aspergillus niger, Aspergillus fumigatus, Candida species, Penicillium species, and these are the most common. Symptoms such as hearing loss, itching, pain, and discharge from the external ear canal are common. Fungal growth may be observed within the ear canal. Cleaning the ears and application of tropical ointments can lead to a resolution of the infection. So those are eyes and ear species, or eye and ear infection caused by fungus. This is going to be the Penicillium species. So you see, you got the septated hyphae. We've got a brush-like appearance as characteristics. So I mean a brush-like appearance at the end here. Um, so yeah, this is a Penicillium species. You note that these are not dark species. That's because they're not dermatophytes or dermatophytes, dermatophores. So now we're going to go into superficial mycoses. These involve the outermost layers of the skin or hair shafts. So these are going to be our infections of the skin and hair, obviously. And that's because they're composed of protein called keratin. Infections involving the hair are characterized by masses of hyphae surrounding the hair shaft. The hair follicles are not usually invaded. The overwhelming majority of infections are not serious, but skin infections may lead to discoloration. So treatment is primarily for cosmetic purposes. Pityriasis versicolor or tinea versicolor is a superficial skin infection that's chronic in nature. The highest incidence of infection occurs in young adults and most often in tropical countries. The high ambient temperatures, hyperhidrosis, so excessive sweating, hyperhumidity, corticosteroid therapy, and malnutrition are thought to be contributing factors to this disease. The upper back, shoulders, upper arms, chest, and abdomen are the areas most commonly infected. The lesions are described as hyperpigmented or hypopigmented, scaly, macular lesions with mild itching. In someone who is light skin, hyperpigmentation lesions have reddish or brownish discoloration, while somebody who has dark skin is going to be hypopigmented and it's going to be confused with vitiligo, which is a benign skin disease. Treatment includes topical therapy. So the etiological agent of pityriasis versicolor is Malassezia furfur. So unlike other fungi, Malassezia furfur has not been recovered as a saprophyte in nature, nor from animal species. This organism is normal flora of the skin in healthy individuals, and it's been recovered from the scalp and trunk of more than 90% of individuals in one study, and no disease was shown. So microscopic examination, you're going to see a cluster of single budding round to bottle-shaped cells. They could be short, unbranched, hyaline hyphal elements. So the hyaline means that they're clear, they're not dark. It's going to resemble spaghetti and meatballs. With a culture, they're going to look lipophilic, which means they require a source of lipids for growth. So they must add them to an oily, must add oil to our agar surface, and they're going to appear smooth or cream-colored yeast-like colonies. It's important that you note the Malaysia furfur is the etiological agent of pityriasis versicolor because that is an exam question. So here is a skin scraping stain with the periodic acid shift or PAS stain. So you see hyphae with budding yeast and they resemble spaghetti and meatballs. So let's continue on with our superficial mycoses. So superficial phagohyphomycosis means brown hyphae fungal infection. It's a chronic superficial skin infection that is most prevalent in young people, less than 20 years of age, but it occurs primarily in tropical countries, including Africa, Asia, Central and South America. It's occasionally seen in the United States and Europe. What we're going to see is a single brown to black irregularity shaped spot with with a sharply distinct border. Occasionally, an infected person may experience slight itching. The lesion is rarely scaly. Most lesions occur on the palmar surface of the hands or the plantar surface of the feet. Occasionally, we'll see it on the neck or the chest. Factors that predispose individuals have not been clearly defined, and the route of transmission is unknown. The causative agent is Hortea warnecki, and it's one of the Dematiaceae or black fungi. Effective treatment involves administration of oral um, alfulvin. Examination of the skin scrapings using KOH and gentle heat will differentiate lesions from melanoma. The presence of a brown septated branching hyphae up to 5 micrometers in diameter and elongated budding cells among epithelial cells is characteristic. By a fungal culture, we're going to use a Sobard dextrose agar, and we're going to see gray colored, shiny, moist yeast-like colony within 1 week. The colony of Hortea wernicki ages it quickly and it becomes black to black. We're going to, I think that's it. Here is the Horatia species. So the next disease that we're going to cover is white pedria, and it's a fungal infection that is uncommon in temperate regions and occurs primarily in tropics. The nature of the route of transmission and predisposing factors is still in question. This condition involves the hair shaft and hair follicles. I mean, it involves only the hair shaft, so the hair follicles and the skin is not affected. The axilla, beard, mustache, scalp, and genital area are the areas most commonly infected. Examination of infected hairs reveals the presence of white to tan nodules that adhere loosely to the hair shaft and they're easily stripped off. Mycelia grow inward, penetrating beneath the hair cuticle and may weaken the cuticle to the point hair shafts may break off above the nodule. This infection is caused by Trichosporon belgii. So diagnosis by a direct examination is going to show us white to tan nodules that adhere loosely on the hair. Cross nodules show round to rectangular arthrocondita and blastocondita. Diagnosis by a fungal culture, we're going to want to do moist white yeast-like colonies with buttery consistence, darkened to yellow-gray and tend to be drier, heaped, and wrinkled. Under our microscopic examination, we're going to see septated hyphae with rectangular arthrocondita. Next, we're going to talk about black pedria. It's an infection of the hair shaft and it occurs primarily in tropical countries such as Africa, Eastern Asia, and South America. The causative agent of this The causative agent of this is a demacious or black fungus known as Pedreaea hortei, which usually infects only the hairs of the scalp. Exposure to stagnant and river water and frequent wetting of the hair may be risk factors. Hard black nodules that adhere tightly to the hair shaft are not easily dislodged are characteristics of black pedria. The infected hair may become fragile and again, the causative agent is Pedreaea hortei. It's a dematiaceae. I can't say this word for some reason. Demacious fungus. Diagnosis is can be made by a direct examination. We're going to see hard to black nodule with numerous ACI containing 2 to 8 astroscopes, which is the sexual state that causes the infection. It can be seen within cavities of infected hair. Diagnosis of a culture, we're going to see slow growing, darkish green, brown to black, velvety colony. Microscopic, again, we're going to see direct, thick-walled, septated hyphae with intracaliary clamatocondia. So here's a black nodule of the species on a brown hyphae. You note that this hyphae is not clear like previous organism because it's dermatophis. So you see the septate here, thick-walled. The next disease we're going to talk about is dermatophytosis. It describes mild to severe diseases of the keratinized layers of the skin, hair, and nails, but spares the subcutaneous tissues. Infection is not confined to the sternum corneum, and as a result, it's the deeper vascularized layers of the skin, such as the dermis, are involved. Where patients are going to experience pain and itching. It's also known as ringworm or tinea. It's going to have a characteristic skin lesion. The outermost ring-like margin is active, spreading infection. It's going to appear red, scaly, raised, and distinct, and it's a central area of healing. Clinical manifestations are named based on the body part affected. So Tinea barbaei is an infection of the beard or mustache, and it's caused by Tryptophyton varicosium or T. metagophragophytes or Microsporum canis. So it's important mostly here to note the names and the causative agents. If you could remember Trichophyton species, then most of the time you'll have a general um, ability to figure out what species is causing this disease. So think Tinea and then Trichophyton. So you're just gonna have to remember the uh, species. Tinea capitis is an infection of the scalp, eyebrows, or eyelashes. It's caused by Trichophyton tonsurans or T. metagophytes or Mycosporium alduni and Mycosporium canis. We could have Tinea corpus, which is an infection of the body where hair is sparse, such as the trunk, and it's going to be T. rubrium, T. metagophytes, T. tonsurans, M. canis, and epidermal phyton floscosium. Tinea crucis is jock itch of the groin or perianal regions. It's caused by T. rubrium, T. metagophytes, and E. floscosium species. Tinus manium is an infection of the palms of the hands between the fingers. This one is caused by T. rubrium. Here you see a picture of ringworm of the arm or Tinea corpus, and this is due to Trichophyton metagophytes. We have Tinea pedis, which is athlete's foot of the plantar surfaces of the feet and the toe webs. It's caused by T. metagophytes, T. rubrium, and E. flocosium. We have Tinea unigallium. This is an infection of the nails, and it's caused by T. rubrium, T. interdigitale, and E. floccosium. Tinea favis or favosa is an infection of scalp hair and the base of the hair, and it's caused by T. Shaolani. Tinea imbricate is a rare form of Tinea corporeus with a bullseye pattern, so it's T. concentricum. Etiological agents, there's over 20 species of dermatophytes. Microsporidium attacks the skin and hair. Epidermal morphyton flocosium attacks the skin and nails. And trichophyton attacks the skin, hair, and the nails. So as you may have seen in the last couple of slides, there's a couple of different types of dermatophytosis infections, and there's over 20 different species that cause these infections. So I would try to review these slides, especially before your quiz, because a lot of the answers are going to come from those slides alone. So etiological agents, arthroprophylactic species is going to be spread by comb, clothing, or bathing items. Zoophilic species are going to be spread by animals such as dogs, cats, cattle, horses, and etc. Gigliophilic species is going to be native to the soil, and they could also be acquired from infective animals such as pigs, yard pigs. Diagnosis is going to be made using a direct examination. So we're going to clean skin and nails prior to collection. We're going to use a sterile scalpel to scrape the leading edge of the lesion. These scrapings are going to come from the doctor on a slide. We're going to put them onto another slide, use to 10 to 20% potassium hydroxide and heat to clear the scrapings. And then we're going to observe it under a microscope. And if there's a infection present, we're going to see long, branching, thick, walled hyphal elements. And we may also see arthrocondydia. So this is what you're going to see a lot of the times in the medical lab is you're just going to take these skin scrapings from a dermatophyto and you're going to do KOH prep and look and just simply note if you see hyphae present or no hyphae present. It's that simple for us as laboratorians. So some dermatophytic infections of the hair can be identified using a Woods lamp. So species such as M. audiouni and M. canis and T. schuleni fluoresce blue or green or yellow-green under ultraviolet light. And this method has been used in the past to screen large numbers of individuals for signs of infection. Some hair projectants produce characteristic patterns of hair invasion, and these patterns may be beneficial in narrowing down the etiological agents. One pattern that we may see is referred to as ectothrix hair invasion, and it's characterized by a sheath of hyphae and arthrocondita surrounded in the hair shaft. So endothrix hair invasion is characterized by hyphae that grow down along the hair shaft and penetrate inside of the hair. Um, you note the species that we're going to see here. In phavic hair invasion, hyphae surround the hair shaft near the opening of the hair follicle and penetrate the hair shaft, creating tunnels within these strands. These tunnels run parallel to the long axis of the hair. Fat droplets may be seen within the tunnels, but arthrocondita are not produced. We could also use a culture. We can use a dermatophyte test medium, DTM, and it turns red with alkaline byproducts. Under a microscope, we can produce club-shaped, cylindrical, or spindle-shaped macrocondita. Macrocondita can be round or pear-shaped and attached directly to the hyphae or short condylophores. Using physiological or biochemical testing, we could see urease activity. So T. metagrophytes is urea positive. We could see carbohydrate assimilation. T. rubrium is able to utilize sucrose. In vivo hair perforation tests. So we could take sterile hair and a sterile distilled water, and its metagrophytes is going to produce a wedge-shaped perforation perpendicular to the long axis of the hair. Here's a positive hair perforation test. See the perforations here in the hair? We could use physiological and biochemical testing such as rice cultures, and it's used to differentiate M. audiolani and M. canis that do not produce macrocandida. So M. canis grows well with yellow to yellow-brown colonies. We could look at colony morphology, so 6 days to 2 to 3 weeks for growth. Flat, fluffy, white, tan, or pink colonies are going to be produced, and it may produce red or yellow diffusible pigments. Here's a picture of Microsporum canis, and it's able to grow on polished rice grains. So Trichophyton rubium typically produces small tear-shaped microcandida on septated hyphae, but rarely macrocandida. Macrocandida are produced in strains that are highly sporulating and are long, narrow, thin-walled, containing 2 to 8 compartments. They're often described as pencil or cigar-shaped. Microcandida may form on the macrocandida. These isolates produce slow-growing colonies which mature in 14 days. On Sobards auger, the surface is typically white to buff in color with downy to fluffy or granular texture. Reversed is deep red or purple but can be brown, yellow, orange, or colorless. And we're going to see red pigment production on cornmeal auger. Here's Trichophyton rubium. So you see the small tear-shaped microcondidia. Next, we're going to cover subcutaneous mycoses and their fungal infections involving deep layers of the skin and subcutaneous tissues resulting in cellular mediated immune response involving lymphocytes, plasma cells, eosinophils, macrophages, and neutrophils. They primarily plant pathogens, but they gain access through trauma and they can disseminate into multiple organ systems. Chromoblastomyces follows a traumatic implantation of dermatious fungi through minor trauma, such as small cuts or puncture wounds. This is a chronic infection that develops over many months with slow progressive lesions that occur primarily on the lower extremities, such as legs and the feet. The characteristic lesion begins as a small, scaly papule that gradually increases in size. If it's left untreated, it could show large raised cauliflower or wart-like lesions, and satellite lesions will develop since spontaneous remission usually does not occur. The most common etiological agent is Fonsecacia pedrosi, and it's the most common worldwide. Other species is Phyllophora vercosea and Claudiophiliaphora carioni. Diagnosis by direct examinations, we could see dermatious septate branched hyphae, presence of sclerotic bodies in diagnostic, mucoform cells that divide and resemble brick wall in the arrangement, thick-walled chestnut brown or copper-colored, spherical oval in shape. See here you see the sclerotic body seen in hemotoxylin and eostain section of a tissue sample. See these yeast-like sclerotic bodies there. So diagnosis using fungal cultures, they're slow growing, 7 to 10 days. The colonies are heaped and slightly folded with a gray, dark olive color, black with velvety surface, and then we have a jet black reverse growth. So with chromoblastomyces, we need to do fungal cultures to diagnose, since all these species produce uh, uh, dermatasis. I can never say this word. Hyphae with sclerotic bodies. So they're often growing in 7 to 10 days. Macroscopically, they're heaped slightly folded with gray. Um, the Cladiosporum species are going to be very tree-like with short chains of oval blastocandida on thick condylophores. Non-pathogenic and must be differentiated from Cladophilioforia carinoni. Its colony is deep olive to black with black reverses. Cladiophiliophora carani is a long, delicate chains of blastocandida that look like they emerge from the hyphae. Colony is gray to green, black with black reverse. Here's a picture of the Cladiosporum species. There's long chains of oval candida and here's the Cladioforia cariani. So you see long chains of blastocandida, indistinct clandidiophores, and no shields. So phagiohypomycosis is a fungus infection that's also caused by dermatious fungi, but they're different than coroblastomyces. Exophilia gene Salamini is the most common isolate of the genus. It shows slender condylophores called anneals and extrude candida into the environment. Annelids tend to taper to a point with cluster of the oval candida. Candida may be seen in clusters alongside the condylophores. Early growth appears as dark pigmented candida only. So here's Exophilia gene salmonelli. It produces septated hyphae, so you could see the septates slowly here. And we have slendocondylophores with clusters of oval candida at the end there. I know at this point you guys are thinking, oh my God, this is really intense. But again, when you go through and study up for the exam, I highly recommend that you just focus on the disease states and what fungi may cause that disease state. So mycetoma is a chronic fungal infection characterized by localized, swollen yet painless granulomatous lesions on the feet and lower legs that can affect hands and arms. Supraputive and draining abscesses are common. Discharge contain colony granules that can be white, yellow, red, or brown and involves cutaneous and subcutaneous tissues but could be spread systemically. Mycetoma refers to a chronic fungal infection characterized by localized, swollen yet painless granulomatous lesions. They're found in the tropics and subtropics. About 50% of cases are bacterial due to aerobic acetinomyces. It's also known as acetinomycotic mycetoma. Could be caused by Nocardia species, Streptomyces species. You guys may remember these. We'll just skip over these organisms because they're not pertinent to the exam. So with a direct examination, we're going to want to examine pus or biopsy for presence of granules. Can be visible to the unaided eye. Crush granule and observe intertwined masses of septate hyphae. Diagnosis using fungal culture. It could take up to 6 weeks to grow. Colonial morphology of Pseudoscariboidea is moderately rapidly growing, produces colonies in 7 days. Initial white with a colony texture that darkens to a grayish brown. Reverse is initially white and eventually turns gray or black. Microscopic morphology. I think let's just skip this slide. Here you're going to see thin, delicate condylophores end with a large single condylium. I'm going to skip these because they're not pertinent to the exam. All right, sorry guys, I had to take a little break. Called a few of my colleagues and asked how important mycology really is because I'm really struggling to get through these lectures, as you guys probably can tell. But um, these end up being board questions, so what I did is I took a lot of the questions from the board of certification book that are tailored towards MLTs and added them to your quiz and your final. And I tried to focus the lecture to where you could find them in our lectures. So it's important. Almost all the information for the quizzes can be found in these slides for these lectures. So at the very least, you could download this lecture and page through it and highlight important information to get through the course. So sporotrichosis, it's also known as Rose Gardner's disease, and it's a fungal disease with worldwide distribution. Most cases occur in temperate to tropical climates. This disease ranges from a local cutaneous infection to a disseminated or systemic infection, and the causative agents is Sporotrix schienecki. You'll have to forgive me for my pronunciation. This is a dimorphic fungi found on plants and soil. So if you know, remember what dimorphic means, it means it has a yeast form and a mold form. It's an occupational hazard for gardeners, florists, and plant nursery workers. Trauma associated with plants, so thorns. If somebody gets caught with a thorn, it could um, help this fungi invade the host tissues. And the most common form of sporotrichosis is the lymphocutaneous form. So in these cases, the initial lesion is small, erythematosus, a relatively painless nodule that forms at the point of entry, usually on the hand or forearm, but occasionally on the feet or the legs. So here is sporotrichosis. It occurs after the spores are implanted by a thorn or a splinter. Diagnosis. So again, it's one of the dimorphic fungi. So it exists as a yeast at 35 to 37 degrees Celsius or as a mold at 25 to 30 degrees Celsius. We could observe yeast in tissues and molds on a culture. It's going to show oval and fusiform budding yeast, often called cigar bodies. You're going to want to remember that. So here are the cigar bodies that you'll see. This is the yeast form. So now we're getting into this really important stuff. So some fungi have the ability to disseminate or spread from the port of entry to infect multiple organ systems. So an example is the fungus may gain entry into the host by inhalation of the candida, and then they're going to multiply within the host and spread to other tissues, such as skin or central nervous system through our bloodstream. So organisms that have low pathogenicity, such as Aspergillus fungum fumigatus, it can end up causing a life-threatening illness in an immunocompromised host. In other cases, organisms such as Sporotrix schienecki, it usually can cause a localized infection and may disseminate into the internal organs such as bone or the central nervous system. And then the most common that we hear about is Candida albicans. It's a yeast and it's found as part of the normal flora of our mucous membranes, but it can also spread and cause multi-organ infections. The four classic agents of systemic mycoses are strict pathogens rather than opportunistics. These organisms include Histoplasma capsulatum, Blastomyces dermatitis, Coxoides immunis, and Paracoxoides brasiliensis. They share the common characteristics to justify studying the organisms in these groups. Um, So let's get into that. I think that's on the next slide. Nope. Let's go back. So these organisms are able to cause disease in otherwise healthy individuals. They tend to be endemic into particular geographic regions. They exhibit dimorphism that is temperature dependent. Their primary target organ is the lungs, from which they may spread to secondary organ systems. So histoplasmosis is also caused Darlington's disease, Darling's disease, cave disease, and Spunkler's disease, and reticuloendothelial and it's a disease of worldwide distribution. It's found in temperate, subtropical, and tropical regions of the world. In the United States, we will see this in Mississippi, Missouri, Ohio, and the St. Lawrence River Valley regions. The etiological agent is Histoplasma capsulatum. It grows best in soil enriched with feces of birds, especially chickens and starlings and bats. Birds are not infected, possibly due to their body temperatures are higher than that of humans. And bats are naturally infected and have intestinal lesions containing the yeast form. With human infections, they may occur when individuals engage in activities such as cleaning chicken coops, attics, barns, and other roosting areas, demolishing old abandoned buildings, and exploring caves. Um, it's estimated that 500,000 people are exposed each year in the United States. In immunocompetent individuals, the immune system is able to contain the mold. About 95% of the infections are asymptomatic, self-limiting, and infections in immunocompromised hosts and are likely due to light exposure of the fungus. Heavy exposure in uncompromised individuals will result in illness that resembles the flu. We can see necrotic lesions in the lungs that become encapsulated by fibrous material and may be visible on radiographs. Immunocompromised individuals are at high risk of developing disseminated histoplasmosis. We could do serological testing for diagnosis. Patients are gonna, there's a complement fixation test and it measures the antibodies directed against this organism. It's the most standard serological test to diagnose histoplasmosis. A titer of greater than 1 to 32 indicates an active infection, and it can cross-react with other fungal agents. We can also do an immunodiffusion test using precipitating antibodies. This is less sensitive but more specific for the diagnosis of histoplasmas. What we're going to do here is we're going to test histoplasmin against sera containing reactive antibody, and what we're going to look for is precipitation bands where antigen-antibody combine in optimal portions. We could also do nucleic acid probe testing, which is probably the most common testing done. Most labs are going to send out any kind of mycology testing. We could also do a direct examination, so we'll see small intracellular yeast in infected tissue. We could do bone marrow and peripheral blood and staining with Giemsa and Wright stain. We'll see intracellular yeast in the cytoplasm of phagocytes. We're going to use chloramphenicol and gentamicin to inhibit the growth. This is an intracellular yeast on a Wright stain of peripheral blood, so you could see it there inside the cell. We could also culture these organisms, so the colony will grow at 25 to 30 degrees Celsius, and it could take up to 8 weeks to grow. We're going to see white to brown with fine, dense, cottony texture. We could see them microscopically at 25 to 30 degrees Celsius. They're going to appear smooth and spiny, round to pear-shaped microcondylida produced on short condylophores or directly on the hyphae. We're going to see large, thick-walled macrocondylida as the colony ages, and it may demonstrate dimorphism. So here we see the mold form. You see a large, thick-walled tuberculate or spiked macrocondylida here. They're spiked. Blastomycosis is another is another systemic disease that we're going to cover. It's also called Chicago disease, Gilchrist disease, and North American blastomycosis. It's a disease of worldwide distribution, and majority of cases occur in North American continent. So we're going to see this primarily in Ohio, the Mississippi River Valley regions. We could also see in St. Lawrence and Arkansas River valleys, Minnesota, and southern Canada to a lesser extent. Dogs and horses serve as additional hosts for the organism. The causative agent is blastomyces dermatitis. What we're what's going to happen is we're going to inhale the candida, and it's going to cause a pulmonary disease, and it can spread to other sites and organs. We're going to diagnose using serological testing. Colonies can grow at 20 to 30 degrees Celsius and are going to appear white and cottony with tan reverse. We could also grow them at 35 to 37 degrees and they're going to appear cream, tan, and waxy. We're going to see septate hyphae with small round condida on short condylophores and directly on the hyphae. In a warmer temperature, we could see large spherical double-walled yeast with single broad-based blastocondidium, and it may demonstrate conversion to the yeast phase. The next disease we're going to cover is coccidioidomycosis. It's also known as Swan-Joaquin Valley fever, desert fever, desert rheumatism, or Posada's disease. It's endemic to the southwestern United States, especially in Arizona and California, northwestern Mexico, Central and South America. The etiological agent is Coxoides immitis, and it's an organism that thrives in semi semi-arid climate where rainfall is low, summer temperatures are high, winters are moderate, and growth is supported by alkaline, sandy soil. It can cause an influenza-like illness that can disseminate, and how somebody would get infected is they're going to inhale the arthrospores from the soil. It affects farmers, construction workers, and archaeologists primarily. We could do two precipitation and complement fixation tests. They're widely acceptable and reliable methods. A high titer exceeding 1 to 16 and persistent or rising titers are indicative of disseminating disease. Excuse me, we could also do a direct examination, such as a KOH preparation of sputum or abscess material and histopathological sections of infected tissues. Um, Here we're going to see spherules that contain granular material or endospores. We could do a culture. It's rapidly growing. It grows in 3 to 5 days. It requires a biosafety level cabinet of 3. No slide culture should be made to observe microscopic structures. It could have a parasitic or yeast phase. It does not behave like other dimorphic fungi. It grows at 35 to 37 degrees, does not induce conversion to yeast phase. Let's keep going. The mycelial colony is going to be initially moist, grayish, and membranous. As it ages, white and cottony texture and may progress to tan or brown. Microscopically, arthrocondida alternate thick-walled, hollow disjuncture cells, 90-degree angle branching, septated, and hyaline hyphae. Paracoxodidolmycosis is also called South American blastomycosis because it's distributed. Um, Distribution is restricted geographically to Central and South America. The etiological agent is Paracoxoides brasiliensis. It usually affects males because estrogen is an inhibitory effect. That's something you're going to want to note about Paracoxidal brasiliensis. It can cause a pulmonary infection, but it could also be disseminated. Here we're going to look at the sputum or draining pus. Peripheral budding around a large spherical double-walled blastocondidium parent cell. It looks like a ship steering wheel or a pilot's wheel. Colony is going to grow at 25 to 30 degrees Celsius, and it's going to appear white with short mycelium, and it turns brown with age. So microscopically, it could be seen at 25 to 30 degrees Celsius. We're going to see septated branch hyaline hyphae with intercalciary or terminal clam, chlamydocandida and a few microcandida along with the hyphae. Colonies are going to appear cream to tan, moist, and yeast-like. Um, we could also see at microscopic at 35 to 37 degrees Celsius large, round, thick-walled cells with single and multiple buds. Buds are attached by narrow connections called denticles. So here is the yeast form of the Paracoccoides brasiliensis. You see buds that form around the central yeast, so it looks like a ship's wheel. The next disease we're going to talk about is systemic penicilliosis due to Penicillium marineffi. It's endemic to South China and Southeast Asia, where it is the third most common cause of disseminated opportunistic infection in AIDS patients. The etiological agent is Penicillium marineffi, and it displays a typical Penicillium mold morphology at room temperature, but a yeast morphology in tissue. The primary source has not been determined. Its most differentiated form, common laboratory contaminants. So Penicillium marineffi, it grows at 25 to 30 degrees Celsius. It's going to appear small, round to oval, non-budding yeast cells with cytoplasm within the cytoplasm of phagocytes, or on direct examination, blood, bone marrow, and skin disease. Rapid produces colonies 3 to 5 days. Flat, powdery to velvet, and maybe bluish green to reddish yellow and yellow or white periphery. Unique soluble red to maroon pigment diffuses into the agar, and the reverse is brownish to red. At um, at 25 to 30 degrees Celsius, it's gonna grow rapidly. Colonies are gonna We're gonna see hyaline septated hyphae with smooth condylophores with 4 to 5 metulate that support 4 to 6 phyllodates with chains of smooth or slight rough candida. At 35 to 37 degrees on enriched media oval, we're gonna see oval yeast-like forms develop. I think there was a few duplicated slides there. I could be wrong. But Penicillium marineffi produces hyaline septated hyphae with smooth condylophores possessing 4 to 5 medullate. So here we see that. It's kind of hard to see the septated hyphae here in this photo. So now we're going to get into opportunistic yeast. Yeast are the most common fungi isolated from human patients, with two species, Candida albicans and Cryptococcus neoformans being the major yeast pathogens. Other species are becoming an increasing problem, particularly for immunocompromised patients with cellular and humoral immunodeficiencies. Um, it's an increased problem with immunosuppression and antibiotic use. They are part of the normal flora of mucous membranes, which makes it more difficult to determine significance, and they recover in large amounts or form a sterile body site are considered significant. So cryptococcosis, a lot of the times when you are working in a lab, you're going to hear an older tech refer to it's either gonna it's Candida or it's Cryptococcus or it's Aspergillus because those are the most common isolates that we're gonna see in a clinical laboratory. So cryptococcosis is a fungal infection that may cause acute or chronic infection in humans. Like any other systemic fungal infection, the primary infection is acquired by inhaling the etiological agent Cryptococcus neoformans, which leads to a pulmonary infection that may be spread to other body sites. Cryptococcus neoformans is known to have a preference for the central nervous system and is spread via the bloodstream. It's a leading cause of fungal meningitis and brain abscesses. Dissemination in other areas of the body can happen, such as skin, bones, and joints. It's able to produce a systemic disease in an individual who have no apparent underlying immune or metabolic defects, but it's most commonly associated with predisposing conditions such as HIV. Signs and symptoms of cryptococcal meningitis may include fever, headache, nausea, dizziness, irritability, impaired memory, and eventually gait and ataxia. Mucopolysaccharide capsule is antiphagocytic and it appears as a halo on stain smears or on India ink agar. The colonies are large mucoid colonies that can be cream, tan, yellow, or pink. Often only a single bud is attached to a parent cell. Diagnosis can be made using serological testing, so we could demonstrate a detectable capsule antigen in body fluids. So we're going to want to do a latex agglutination test here. A serum titer of 1 to 4 or higher can indicate an infection. Titers of 1 to 8 or greater indicate an active infection. And we're going to use the titer to monitor therapy. So if the therapy is working, that titer should lessen. We could also do a direct examination. So we're going to do India ink to demonstrate the capsule in body fluids. The ink cannot penetrate the capsule, so only about 50% positivity rate, so negative preparations must be cultured. It's going to show spherical, single, or multiple budding yeast cells. We could also diagnose from cerebrospinal fluid, blood, or urine cultures. Macroscopically, they're going to appear large, tan, or mucoid. We're going to use Niger seed or birdseed agar, and that's going to result in a dark brown to black colony. The organism produces phenol oxidase that converts phenolitic compounds to melanin. It's available as rapid disc caffeic acid. Other biochemical testing include rapid production of urease. The next infection we're going to talk about is candidiasis, and it refers to any infection caused by a member of the Candida genus. Candida species such as Candida albicans, Candida tropicalis, Candida parapsilosis are responsible for a wide variety of illnesses, from chronic cutaneous and mycocutaneous infections to fatal disseminated disease. And people who use antibiotics, people who are diabetic, and phagocytic deficiencies can trigger the disease. There can be part of the normal flora and mucous membranes, and we're going to want to treat it with nystatin, azoles, and amphotericin B. The presence of blastocandida in a specimen from a non-sterile body site, such as a mucous membrane, may or not be significant because yeast may be colonized in the body sites, including skin and mucous membranes. So without clinical symptoms, we cannot know if an infection is actually present. Um, so it may or may not be significant. We could grow on a routine culture using EMB agar or McConkey agar and fungal media. Here we're going to see a small oval budding blastocandida, and the long pseudohyphae can be seen on the gram stain. Smear of a vaginal secretion. So we see the small blastocandida with the long pseudohyphae here. It's most likely Candida albicans, and in some cases it could be a sexually transmitted disease. So when you do a wet prep or a well, most likely a wet prep from a vaginal specimen, whenever you see a budding yeast or branching yeast, it's more than likely going to be a Candida species. We could use assimilation studies or fermentation of carbohydrates or production of urease to diagnose. Candida albicans can be an isolate from the upper respiratory, gastrointestinal, or urogenital tracts. It causes thrush, diarrhea, UTIs, vulvovaginitis, and catheter-related blood infection. Thrush is probably the most common infection associated with Candida albicans. Here we see Candida albicans and a patient with thrush. The colony is going to appear white to cream colored, smooth, glistening colonies in 3 days. It can have extensions around periphery of the colony. It could appear as feet or fringe appearance. In patient specimens, blastocandida, pseudohyphae, and true hyphae are present. On CMA, blastocandida, pseudohyphae, and true hyphae, and we can see chlamydocandida. I think we'll skip the first part. Um, but Candida glabrata causes infection primarily in immunocompromised individuals. It's part of the normal flora. Pneumocystis jirovecii infection. Pneumocystis jirovecii. I guess we're skipping past the Candida species, and we're gonna go right to Pneumocystis jirovecii. It used to be considered a parasite. When I went to school, Pneumocystis jirovecii actually was a parasite. Um, it could be visualized using methamine silver or toluidine blue or game sustains. Causes interstitial plasma cell pneumonia. It can happen in premature or malnourished infants or older adults, immunocompromised patients, especially AIDS patients, and it has a high mortality rate. We're going to treat Pneumocystis jirovecii with trimethoprim and sulfamethoxidol. Here you're going to want to remember Pneumocystis jirovecii with AIDS. Not be cultured on artificial media. Histological staining is used for diagnosis. We're also going to take respiratory secretions, washings, bronchial lavages, and lung tissues for histological staining. GMS, PAS, and cauliflower white stains. Stain the spherical cyst wall only. Shows cup-shaped cyst, gray to black in a foamy exudate, green background on GMS. Games on white stains, we could stain the intercystic bodies and trophic form, but not the cyst wall. Geotrichosis is caused by Geotrichum candidum. Can be part of normal flora. It's found on fruits and vegetables. Can cause wound infections, thrush, skin lesions, infected nails. Rapidly growing, produces white to cream yeast-like colonies. We're going to see septate hyaline hyphae that fragment into one cell rectangular arthrochondrida. Here's the Geotrichum species. You could see the septate a little bit. We're going to see hyaline hyphae, so those are the clear hyphae, and they could fragment off into one cell rectangular arthrochondrida. So this area right here could actually fragment off. Now, the next few slides are just plates, and these plates are going to be coming from the book Bowman's Colored Atlas. So I'm just going to briefly go over them, but it's important that you guys take your atlas and go through these pictures so that you could get a good visual idea of what these organisms are, because you may simply just get a visualization and clinical symptoms and need to know what fungus is causing the disease. So here we're going to see zygomycete colonies rapidly fill the plate, gray to brown in color. It looks like pepper. Here's the hyphae of zygomycetes. It looks ribbon-like. This is a Rhizopus species. It's the most frequently encountered pathogen. You see that rhizoids at the base, and we have already colored this, so it looks kind of like roots. Here's an invasive hyphomycosis. This is Aspergillus. This is Aspergillus fumigatus, and it produces blue-green colonies. You've seen that in an earlier slide. Here's Aspergillus the hyphae form. Here's Aspergillus niger, which is the mold. Produces long condylophores and commonly produces fungus balls in infected patients. Here's Penicillium. We'll skip this one because we never covered it. Here's the fusiform species. So you remember it kind of looks like a boat, crescent shaped. This is a Pseudodalsyriboides. Here's Microsporium. You're going to note that macrocondita at the end there. This is a Cryptophotian species. So you remember these are the ones that cause your skin, hair, and nail infections. Dermatophytes, you notice that they're darker. Here's Epidermorphotone species, Flossicosium. Remember we talked about this again with our tinea diseases. Here's Blastomyces dermatitis. It's a large yeast. Here's Coxoides species. Histoplasma capsulatum. Kind of looks like a wheel there. Ship wheel. Here's Curvolvera. And this is the Candida albicans. This is the one you're going to see the most often in the clinical lab it's a budding yeast. This is a germ tube test. You're going to want to remember germ tube testing and Candida species. Here's Cryptococcus neoformans, capsule producing yeast, and it's a human pathogen, especially in immunocompromised patients. You're going to want to remember that. And that's it. I apologize that this lecture wasn't amazing by any means, but bear with me. Mycology is one of the hardest areas of the lab, and as long as you could get the general names down, a visual representation of the important or pertinent bacteria, which is the last 10-ish slides here, I think you'll be fine. If you know that Cryptococcus, Candida albicans, Fusiform, Penicillium, Pneumocystis jirovecii, I think you should be fine as far as my quizzes go and hopefully for the boards. Happy studying.